Okay, I'm going to be talking about the response of Natalgrass, Melinis repens, an invasive species in Florida scrub vegetation to supplemental additions of nitrogen and phosphorus. Okay, this is a map showing the distribution of Natalgrass. Historically, it apparently was in southern Africa, and now it has a much wider distribution. If we look at, this is a picture of the grass, Melinis repens, and it initially apparently was brought in to Florida at least because it was a plant they could use for escaping the landscaping, and it is in other states as well. If you look at the upper map, you can see that the southern states many of them are included in the Natalgrass. And then if we look into Florida, most of the areas are at least experiencing the tall grass. Okay, what we're going to do here is I'm going to talk about how nutrients in the soil are going to affect the growth of the plants. Okay, and if you look at the soil that's available, the nitrogen, we're just going to be working with nitrogen and phosphorus. Nitrogen is the ammonia form, and it had 0.8 microgram per gram of soil. And for phosphorus, it was 1 microgram for soil. Okay, and so the soil doesn't contain very much of those two nutrients. We did two different studies. One is what I call the pot culture study. And the pot culture study, we field collected soil down from 0 to 15 centimeters on May 2nd. The pots contained around 1,600 cubic centimeters of soil. The seedlings were planted on May 3rd. We had 15 replicates for treatment. There were 4 treatments: nitrogen only, phosphorus only, nitrogen and phosphorus, and water only. There was a pre-treatment measurement time, and then we had the first application of the treatment. So the first treatment measurement was to measure the plants that we were working with. Okay, this shows the pots that are there, and we had them on 3 benches, and then they were just randomly located, and we moved them around so that they would locate different portions of the bench. The treatments that were again nitrogen, water, phosphorus, and nitrogen together. There were 4 applications. Nitrogen had accumulated 20 micrograms per gram. Phosphorus, it was 19 micrograms per gram. Okay, if you look at this one, you can see when the application, back up one. If you look at this one, you can see that the total number of days that the plants were treated with nutrients. So there was 4 days. With the case of phosphorus, each of the days counted for 3 micrograms per gram for the total of 12 micrograms per gram. For nitrogen, we had 5 micrograms per treatment for a total of 20. The N and P combined the phosphorus and the treatments together. Each application added 125 milliliters of water to the treatment. Okay, this is looking at 2 aspects. One is the soil, the log total dry mass. This is at the end of the experiment when we're getting plants weighed. And then we have what we called an index value. Okay, the log biomass was plotted over the index, and the index, what we did is we counted the number of stems which were less than 10 centimeters in the plant, plus we had stems which were taller than 10. We multiply those times 4 because they're more representative of how the plant was growing, plus maximum height. Okay, this is the biomass for the treatments. N and P had the most. The other 3, phosphorus, water, and nitrogen, were much less, significantly less, and they I'll be showing you more of that in just a minute. Okay, this is looking at the distribution again of the plants. This is the dry weight in grams, and this is the index. Okay, if you look here, the NNP has the most distribution, the highest level of weights and index value, and then the other remaining ones were pretty much all the same: the nitrogen, phosphorus, and the water. This is looking at changes in growth index, and if you look at them, we measured the growth a total of 4 times: May 9, 24, June 15, July 9. And you can see this is when we first started. There was no difference. Okay, but as the plants grew, and particularly the NNP, they had higher levels than any of the other treatments. Phosphorus was a little bit higher, and sometimes with respect to the other ones, but it was not as important. Okay, so that was the pot experiment. This is the field nutrient experiment. We had a total of plants for treatment, and the plants, for some of them, were getting nitrogen and phosphorus, and that was the number of 24 pots. We had water 25 and control 25. Okay, in this case, we didn't add simple nitrogen and phosphorus separated because it's a little complex to do that in the field. Okay, the nutrient addition, we had 1 liter of water, 1 liter of solution or water per plant at each application. Okay, if we look at the application days, the days that the plants got nutrients, and this would be for the N and the P, it was March 20, April 21, and May 19th. Okay, this is the way it looked like when we were adding the nutrients or the water to the treatments, the plants. The little metal box here is 25 centimeters or 10 inches in the sides, and this would be this is used to hold the water or the nutrient in place. Okay, then we had monitoring days, and these are the days when we measured the plant using, as you'll see later, the same index that we did otherwise. Okay, so the pre-treatment time was March 14, and then March 31, April 22, May 2nd, May 20, July 17 and 12, and August 18 and September 22. The experiment duration period of time was 185 days after the first application. Okay, so we've got the first application shown. Okay, what we did then is we computed a growth index, and it's the proportional change in growth. And what we're looking at is the growth index at time, and there were a total of 7 times, or actually 8, when we were measuring the data. The first time was when we used just pre-treatment at the growth index, and then it was multiplied times 100, and then pre-treatment growth index. Okay, the pre-treatment growth index is the growth that occurred on the first time of measurements being made, and then it resulted in trying to separate or to measure the individual times that it happened. Okay, so if you look at the number of times, this is IT. Okay, it varies from a total of 7 monitoring dates. Okay, the pre-treatment growth index is calculated by the number. Well, I can't really see this too well. Pre-treatment is calculated by the number of stems less than 10 centimeters, 4 times the number of stems greater than 10 centimeters, plus the maximum height for the measurement of the plant. Okay, so this is taking place between this and this. Okay, the growth is the one which is going to be changing excuse me, the pre-growth index treatment is going to be separating, subtracting the treatment that it has, the grow index and so on. But it doesn't happen at a rapid period of time. Okay, so it's I here, the growth index of time. It's a result of the growth index of time varies the monitoring dates from 2 to 8, and there were a total of 8 treatments, 2 to 8 from the index, and then we had one more measurement that we made, but that was followed by giving the first treatment. Okay, so if we look at the results, what you can see, excuse me, is an axis here which has the log proportional growth index. Okay, and then this one is the days since the number of initial nutrient addition. Okay, and so this would be the first one, the second, the third, the fourth, the fifth, the sixth, and the seventh. Okay, but there was a total of 8, and that first one was not included in this pattern. Okay, now if you look at what's happening, is that in the first few treatments, there were no significance between them. Okay, so this was for treatment 2, 3, 4 was significantly different than all the other 2 treatments, the water and the nothing. And then the next one is looking at treatments which are significantly different for the green here and the blue. Okay, and then the red one was B, so there's no significance between them, difference. But then if you look at this one, the next 2, there's no significant difference in between them. Okay, now if you want to look at this, I can tell you probably why it's happening. And if you look at the second to the last one, they had a precipitation for that particular month, was in August, of 220.05 inches of precipitation. Okay, the second one is the amount of precipitation that occurred between August 1 and 18. August 18 is when we made the measurements. Okay, and so what's happening, I think, is that the rainfall is very high, and the nitrogen and the phosphorus treatment is going to get washed out. So I would expect that without that problem, there would be significant differences. So I think that's the last of my questions. Yes. Okay, conclusions. Both nitrogen and phosphorus are limiting nutrients for Natalgrass grown in soil from scrub vegetation sites. Increased growth occurs only when both nutrients are provided. And that's what I have.